Okay, well, welcome again. So this is the second video in the series of this particular cell paper that we're going over. So if you haven't seen the first one, please go see that one first. And if you have any questions, just leave them down into in the comments. So, so this is where we left off, was getting into the architecture of the Sars CoV-2 S-glycoprotein trimer, specifically the portion of the trimer that is outside of the virus particle. So, but before we go into that, there is just one amendment that I wanted to do to the last video regarding the kinetics of the spike protein interacting with the ACE2 receptor. So this is in the biolayer interferometry binding analysis, and these curves that you see, these graphs show the binding of the ACE2 receptor to the spike protein portion, and then having it come off. So down here we have some of the calculated constants of this particular assay, and it might get a little bit too mathematical, but the things that you need to just get out of this is that when it comes to this small k on, this is the rate at which the particles are coming together. So this is basically the speed at which the spike protein and the human ACE2 receptor, if they're floating around in the solution, it's the speed at which they bind. And so here, it's hard to see it because the graphs are scaled differently. But here, the speed is pretty much equal. The velocity is pretty much equal. So here you can see the K on is 1.4 times 10 to the fifth for the SARS coronavirus 2 spike protein, and it's 1.4 times 10 to the fifth for the SARS coronavirus original spike protein. So as you can see, between them, pretty much identical as far as how quickly it starts to, or how quickly they're coming together. Now, but just because they start off quickly doesn't mean that they're going to end at the same concentration or the same amount being bound. So as you can see right here, even though they start off at the same speed, the original SARS spike protein ends up, reaching its equilibrium close to around, well, it looks like it'll be around 0.3.5 for the new spike protein, or the new coronavirus spike protein. It looks like it goes all the way up pretty much to 1. So even though the rates at the beginning are the same, they end up reaching their equilibrium at completely different concentrations. So that is for the K on, but the K off, the K off is is this part of the graph right here, where they end up getting dissociated and coming off of the receptors. And so here you can see for the new SARS coronavirus, that process is slow. For the old SARS coronavirus spike protein, that process is quick, or much faster relatively. And so I should also mention that it is slower, coming off of the receptor is slower than it is attaching, and that you would expect to see that because usually when the two particles end up coming together, there's usually some sort of conformational change, or the proteins change shape a little bit, and by changing shape, it locks that position and make a little bit better. So coming down to the table over here, we can see that the K off for the original SARS coronavirus spike protein is 7.1 times 10 to the negative fourth. And just, these are the units for these K values, but it's 7.1 times 10 to the negative fourth. However, it is only 1.6 times 10 to the negative fourth for the new SARS coronavirus spike. So this is telling you that if given a particular concentration of bound receptors, the old coronavirus can get, if it's being washed away, will come off of the receptor about 3 times more like 4 times more easily than the new SARS coronavirus. So just keep that in mind. So not only does not only does the new coronavirus spike protein, SARS-CoV-2 spike protein, not only does that bind better, but it also comes off of it a lot more slowly. So continuing on to the architecture of the SARS coronavirus 2 glycoprotein trimer. So here's some nice photos. And here you can see that with this technique that they use to help image this spike protein, that they discovered that there are 2 general trimers that they find. So they find the closed SARS coronavirus as trimer, and here, this is the binding domains. The binding domains that we were talking about, which is the part that sticks to the ACE2 receptor, here, they're all closed. So all 3 binding domains are hidden, tucked away within the trimer. So they discovered that this is one conformation, and then the partially opened one is another conformation where you only have one of these binding domains released. So the partially opened one is the other general structure that you find. Now, granted, these are not the original spike proteins of the SARS coronavirus 2 trimer. So they did some genetic modifications to this to come up with these structures. So here we'll continue down. So to enable single particle cryo-EM studies of the SARS-CoV-2 as glycoprotein, we designated a pre-fusion stabilized ectodomain trimer construct with an abrogated furin S1, S2 cleavage site, 2 consecutive proline stabilizing mutations, and a C-terminal fold-on trimerization domain. So these are the genetic engineering techniques that I was talking about. So the first thing that we want to talk about is they remember the furin cleavage site that I was talking about before. So with the coronavirus cultures that they did with this new coronavirus, what they found was there was a protease cleavage site that was very active as far as how easily it was cleaved by the proteases within the cell. And it's so active that as the virus is being produced, the enzyme, the furin enzyme, is already destroying part of this spike protein. Excuse me. So what they had to do in order to have a lot of the same particles to deal with, they had to remove that furin cleavage site. Because if you have a whole bunch of different types of spikes, it'll be much harder to come up with the resolution that you need for this cryo-EM experiment. So they got rid of that. So just keep in mind, this is not going to be the type of structures that you see in the real live virus circulating around, most likely. So that was one of the things that they changed. The other thing that they changed was 2 consecutive proline stabilizing mutations. So I'm not exactly sure what that would mean. I would only assume that they replaced a few of the amino acids into prolines, and by doing that, they've restricted the free movement of the spike protein just a little bit, and that can help prevent, I guess you could argue that it gives better resolution to the entire structure, so it's less floppy, if you will, because prolines are a very restrictive amino acid, and the more prolines you have, the less freedom of movement the protein has in those particular locations, as a general rule of thumb. So they put 2 consecutive proline stabilizing mutations in order to accomplish this. And they also did a C-terminal fold-on trimerization domain. So this is not the actual spike protein, because normally the spike protein has an entire other section that is inside of the virus. So here you can see they have denoted the viral membrane, but this is missing. They removed pretty much half of the, I shouldn't say half, but they removed a lot of the spike protein because they are only interested in the ectodomain, or the outer domain right now. But if you remove so much of your enzyme, it's very difficult to have the original trimerization take place. So not only did they not only did they um, remove most of the protein, or I shouldn't say most, but a lot of the protein, they added a trimerization peptide. So this is a genetic and an amino acid sequence that if you add it to a protein, it's pretty much guaranteed to trimerize with two others of itself. And so by bringing together three individual pieces of the spike glycoprotein, you can re-emulate what the original virus spike protein is going to do. So, and this is good because then you can just take a part of your protein and it's easier to do the algorithms on a smaller particle, or I should say a less variable, smaller part of a protein than it is to do the entire protein all at once. So continuing, three-dimensional 3D classification of the cryo-EM data revealed the presence of multiple conformational states of SARS-CoV-2 corresponding to distinct organization of the S suit binding domains within the S1 apex. So this is at the top, these are the S1 apexes right here. And the binding domain is tucked away over here. Excuse me. So approximately half of the particles half of the particle images selected correspond to trimers harboring a single S2B domain opened, whereas the remaining half were accounted for by closed trimers with the three SB domains closed. A recently determined SARS-CoV-2 S structure also detected trimers with a single SB domain opened, but none entirely closed. The observed conformational variability of SB domains is reminiscent of observations made with SARS-CoV-S and MERS-CoV-S trimers. Although we did not detect trimers with two SB domains open, and the distribution of particles across the S conformational landscape varies among the studies. So what they're saying is there is general reproducibility going on here, but not absolute reproducibility. So these are the proportions that they got. They got about half closed, and they got about half with only one of them opened. Who knows if that's what's actually happening in the real life viruses? But there is some other labs have shown that you do get a variety of open and closed states, such as this. So continuing, we determined a reconstruction of the closed. Oh, actually, what I should do before we continue, I should talk about how they even come up with these structures in the first place. So cryo-EM is cryo-electron microscopy. So cryo meaning very cold, and electron microscopy, where it's basically you shoot electrons at your subject, and then the electrons are moving so quickly they behave like light particles, and then they are able to do imaging based on that. So what they do with cryo-EM is they take a small solution with a bunch of these proteins that they've generated inside of them. And then after they get it really cool, they shoot it with the electron microscope, and then they get an image with a bunch of particles in it. But all of those particles are really blurry because these structures are too small to be resolved with the electron microscope. But if you take thousands of these blurry images and you combine them together with specialized algorithms, and who knows exactly how they work, but eventually you can determine a structure that would cause all of those blurs. And sometimes you have two structures, so you have to be able to separate what blurs go into one category and which blurs go into a different category. But that's basically how it works. So we determined reconstruction of the closed SARS-CoV-2 ectodomain trimer at 2.8 angstrom resolution, and angstrom is just a measurement of distance, and applying threefold symmetry and an asymmetric reconstruction of the trimer with a single SB domain opened at the 3.2 angstrom resolution. So what they're saying is one of the ways to help determine the structure is if you know that there is symmetry going on, and that is an assumption, that's not always the case, but if you know that there's symmetry going on, that can help your algorithms decide what structure you you do have that are creating those little blurry dots on your image. But they also had to account for sometimes you don't have a symmetric structure. So right here, where you have the partially open SARS-CoV-2 S trimer, one of them is sticking out. And they were able to basically tell their computers, okay, what if one of them's sticking out? What images are we going to get from that as well? So the SF2 fusion machinery is the best resolved part of the map, whereas the S-sup A and the S-sup B domains are less well resolved, presumably because of conformational heterogeneity. So conformational heterogeneity would mean that um, so for instance, with the binding domains, um, we would assume that after they interact with something, that they would want to change their shape a little bit so that they can stick better to what they've interacted with. And so that would probably indicate that it's a little bit squishier than some other parts. And the problem is, if things are squishier and if things are moving around, it's much harder to get a good resolution image of those particular locations. So that's basically just what they're saying here. The atomic model comprises residues 27 through 1147 with internal breaks corresponding to flexible regions. So what they're saying is there are some parts that are so floppy that they can't resolve it. So they don't include those portions of this structure. So that's just one of the limitations that we have dealing with our experimentation. So including part of the RBM, so the receptor binding domain, and lacks the C-terminal most segment that is not visible in the map, as in the case for all S structures determined to date. So they're saying we couldn't do this, but neither could anyone else. So, you know, we can't reproduce this because we can't produce it in the first place, and neither can anyone else. So it's just a way to cover your tracks. Overall, the SARS-CoV-2 S ectodomain is a 160 angstrom long trimer with a triangular cross-section, resembling the closely related SARS-CoV-S structure. As is the case for the other beta coronavirus S glycoproteins, including SARS-CoV-S, the S1 subunit has a V-shaped architecture. In the closed S trimer, the three human ACE2 recognition motifs, whose location was inferred based on the crystal structure of the SARS-CoV-SB in complex with the HACE2, are buried at the interface between promoters. So a little bit to unpack right there. So basically they said somewhat V-shaped. It's also got a triangular cross-section. So when you look at it from on top or on bottom, you can see a cross-section of a triangle. And they also argued or could figure out, based on the crystal structures of SARS-CoV-SB in complex with HACE2, they realized that the SB is tucked away. And we should mention crystal structures. So crystal structures are basically, sometimes you are able to crystallize proteins if you can get the concentrations high enough and if you can get the conditions just right for them to crystallize. And then oftentimes what we do with those crystals is we will shoot them with X-rays, and then the light will scatter after it hits the crystal of the protein. And basically, we can look at the light that is scattering off of the crystal, and from that we can run algorithms that can determine what the structure of that crystal is. Or I shouldn't say, the structure of the protein that makes that crystal is. So in the case for the SARS-CoV-2 S binding domain with the HACE2 receptor, they were able to figure out what amino acids are touching the HACE2 receptor. And they realized, okay, those parts are tucked away. Those amino acids are tucked in these closed conformations. But, you know, also there's an open conformation as well. So we would assume that this might be a way to protect the amino acid sequences. So it'll be more difficult to make antibodies against this particular sequence. There's a lot of different ways that a virus can use that strategy for its fitness. So continuing, as a result, SARS-CoV-2 S binding opening is expected to be necessary for the interacting with ACE2 at the host cell surface, and initiating the conformational changes leading to the cleavage of the S2' site, membrane fusion, and viral entry. Okay, it's continuing. As the SARS-CoV-2 and the SARS-CoV-S sub-2 subunits share 88% sequence identity, they are structurally conserved and can be superimposed with 1.2 angstrom mean square deviation over 417 aligned carbon alpha positions. Best way to describe this is that they're saying the SARS-CoV-2 and the original SARS-CoV S2 subunits are very, very similar in their sequence identity. And they can, to superimpose them means that you can take one structure and then you can take the same structure and you can sort of like, if one of them's a ghost, you can put them on top of each other. Excuse me. And what they're saying is the differences between those structures is very small. And if you take the 417 carbon alpha positions and carbon alpha positions, so 2320. So here, this is a amino acid where you have the amino group and the acid group. And these are a part of a polymer in a protein or a peptide. So this carbon right here that is in between the amino group and the carbonic acid group, this is the alpha carbon, and this R group changes depending on which amino acid you are talking about. So when they say over here that you can align 417 of these alpha carbons on both the new and the old coronavirus, and they are pretty darn close together, over 417 aligned residues. So that's basically what they're saying here, that they're very similar. So over most N-terminal, only the most N-terminal part of the fusion peptide is resolved in the map, as well as the case for previously determined SARS-CoV spike structures. The sequence and conformational conservation of the fusion peptide region observed across SARS-CoV-2 spike and SARS-CoV spike suggests that antibodies targeting this functionally important motif might cross-react and neutralize the two viruses as well as related coronaviruses. So the sequence fusion. So what they're saying is the sequence and the conformational conservation of the fusion peptide region observed across these two different coronaviruses would indicate that if you could get an antibody that can work well against this area, it won't just work for one coronavirus, it's going to work for both of them. So, continuing, we previously showed that coronavirus S glycoproteins are densely decorated by heterogeneous N-linked glycans protruding from the trimer surface. So here you can see these N-linked glycans. So glycans are just small sugars. And N-linked means that they are connecting to nitrogens on the side chains of these of the amino acids of the protein. And so here, all these dark blue spots that you see are a different sugar. Now, I'm not exactly familiar if this is just the first subunit or the first monomer of the sugar, or if there's actually longer chains of sugars coming off of them. And I would assume if it's a long chain, then it would be very difficult to resolve the image in cryo-EM. So they're probably just showing the first, if they are long chains or longer oligomers, they're probably just showing, the first sugar on here. So keep in mind, this is a post-translational modification. So this is something that, this is a sugar that your own enzymes are going to produce and put on the virus proteins itself. And it would be a strategy that a virus might use to help hide or protect itself from your own immune system, or be able to hijack some of the cellular machinery to be processed and moved in different ways. So continuing. So these oligosaccharides participating in S-folding affect priming by host proteases and might modulate antibody recognition. So that's exactly what I just said earlier. So SARS-CoV-2, a spike SARS-CoV-2 spike comprised 22 N-link glycosylation sequons per promoter, and oligosaccharides are resolved in the cryo-EM map for 16 of these sites. By comparison, SARS-CoV-S possesses 23 N-link glycosylation sequons per promoter, and we previously experimentally confirmed that at least 19 of them are glycosylated. Just need to double check. Okay, yeah. So a sequon is a sequon is a sequence of amino acids that can give rise to a glycosylation event. And so what they're saying here is for the SARS-CoV-2 S protein, there are 22 N-link glycosylation sequons per promoter. So they would believe there to be 22 N-link glycosylation events going on. And they were able to find 16 of them. So they were able to find 16 of the 22. That doesn't mean that, it could mean that those are the only ones, or it could also mean that they just could not find it at the other locations, but they are actually there. Continuing by comparison, the SARS-CoV-S possesses 23 N-link glycosylation sequons per promoter, and we previously experimentally confirmed that there are at least 19 of them that are glycosylated. So if you can find them, you can guarantee it's there, but that doesn't mean that you found all of them. So 20 out of 22 SARS-CoV-2 N-link glycosylation sequences are conserved in the SARS-CoV-2 specifically 9 out of 13 glycans in the S1 subunit, and all 9 glycans in the S2 subunit are conserved among SARS-CoV-2 and SARS-CoV-S. Furthermore, S2 N-link glycosylation sequences are mostly conserved across SARS-related coronavirus S glycoproteins, suggesting that accessibility of the fusion machinery to antibodies will be comparable among these viruses. So they're arguing that between the original the original coronavirus spike glycoproteins, they have a lot of sequences that would indicate that they are glycosylated, and 20 of them are conserved in the new SARS coronavirus as well. So there's a lot of matching going on. And then furthermore, a lot of other related coronaviruses also have a lot of these sequences as well. So they presume that there's a lot of spillover as far as where these glycosylation events are happening or where these sugars are being placed. And so this would lead credence to the argument that antibodies that work against one are going to work against all of them. So if your immune system is able to figure out how to properly make antibodies for one of them, that same technique will be beneficial at taking care of a different coronavirus. So SARS-CoV-S elicits neutralizing antibodies against SARS-CoV-2S. So mapping of the sequence conservation across multiple spike sequences from the SARBA subgenus underscores that the S2 fusion machinery is more conserved than the S1 subunit, with the highest divergent found in the S sup A and the S sup B regions. These observations are in line with 1, the fact that some, but not all, of these viruses use ACE2 as an entry receptor, and 2, that S1 subunit is more exposed at the viral surface than the fusion machinery and is likely to be subject to a more stringent selection pressure from the immune system. Based on these, okay, so what they're arguing right now is that since the S1 subunit is exposed, what it's probably going to want to do is change more frequently because it's always trying to avoid the immune system. So by avoiding the immune system, you can just change your surface over and over again, and that would be a way to change which antibodies would work against you, or at least make some older antibodies obsolete. But this selection pressure does not exist for the fusion machinery that is more well protected. So if you can create an antibody that can nestle into that well protected area, that will be superior. And since that fusion peptide is really conserved among a lot of coronaviruses, if that antibody works for one, it should work for a lot of others as well, presumably. So based on these observations, we hypothesized that exposure to one of the two viruses could elicit cross-reactive and potentially neutralizing antibodies against the other virus. We therefore investigated the ability of plasma for our 4 mice immunized and stabilized with SARS-CoV spike to inhibit SARS-CoV-2 spike and SARS-CoV spike-mediated entry into target cells. All sera tested completely inhibited, and sera would be like blood or the plasma. So all the plasma tested completely inhibited transduction of SARS-CoV SMLV, which, remember, this is the genetically engineered weakened virus with the original SARS spike protein. So it completely inhibited this from working, but it also reduced the SARS-CoV-2 SMLV. So taking a virus that has the old SARS spike protein, getting the antibodies that were generated from that, completely inhibited this, but then it also inhibited this as well, the SARS-CoV-2 SMLV, the new one. And it reduced it down to 10% of control in these monkey cells. So if you can reduce the virus by a factor of 10, that is phenomenal. And that will basically help your immune system keep the numbers down low enough to fight it off more rapidly. So the elicitation of a heterotropic response blocking SARS-CoV-2 mediated entry into the host cells concurs with the sequence and structural conservation of the SARS-CoV-2 and the SARS-CoV-S, along with their comparable glycan shields, because they're shields, they are shielding the virus from your own immune system, and suggests that immunity against one virus of the SARS coronavirus subgenus can potentially provide protection against related viruses. So what they're saying is a heterotropic response, or basically getting a lot of different enzymes, a lot of different antibodies and mixing them together and injecting them, that ended up working for the SARS-CoV-2 SMLV, or the SARS-CoV-2 spike. So there was cross-reactivity. Entering the discussion, receptor recognition is the first step of viral infection and is a key determinant of host cell and tissue tropism. Enhanced binding affinity between SARS-CoV-S and human ACE2 was proposed to correlate with increased virus transmissibility and disease severity in humans. Indeed, SARS-CoV isolates from the 3 phases of the 2002 and 2003 epidemic were more efficiently transmitted among humans and more pathogenic than the isolates associated with the 2003-2004 reemergence that caused only a few cases in line with their binding affinities for HACE2. So here they're saying with the old SARS epidemic, weak binding meant weak pathogenicity and weak virality, whereas strong binding meant high pathogenicity and high transmissibility. Continuing, moreover, the ability to engage ACE2 from different animal species appears to reflect host susceptibility to SARS-CoV infections and facilitated the jump of the virus from animals to humans. So because ACE2, and keep in mind it not even be called ACE2 if it's found in a different species, but the ability to engage ACE2 from different animal species appears to reflect host susceptibility to SARS-CoV infection. So since this ACE2 is found, or at least similar structures are found throughout the animal kingdom, the virus can pretty easily make the jump from one species to the next, unfortunately. Unfortunately for us. We report here that SARS-CoV-2 uses human ACE2 as an entry receptor and recognizes it with a similar affinity for the 2003 2002-2003 SARS-CoV isolates, which suggests it can spread efficiently in humans, in agreement with numerous SARS-CoV-2 human-to-human transmission events reported to date. I mean, it's an understatement. It binds much better than the original SARS, but let's leave it at that. Besides the binding to host cell receptors, priming of the spike glycoprotein by host proteases through cleavage at the S1, S2, and the S2 prime sites is another crucial factor modulating tropism and pathogenicity. For instance, entry of the MERS-CoV-related bad coronaviruses HKU4 into the human cells requires addition of exogenous trypsin, indicating that S proteolytic activation of the bat virus did not occur in human cells. So what they're saying is when they tried to, not this particular lab, but a different lab, when they tried to infect a line of human cells with HKUs, they really had to add a lot of protease to it for it to effectively work. So this would indicate that this virus, as is difficult to get into human cells. Indicating that S proteolytic activation of the bat virus did not occur in human cells, despite its ability to recognize human DPP4. And if I remember correctly, I believe this is a receptor. Yeah, dipeptidyl peptidase 4 is another receptor that viruses are able to take advantage of. Well, that's precisely the one that the original MERS took advantage of. So, and remember, this is a MERS-CoV related bat coronavirus that is using the same receptor as the original MERS. However, it required a lot of extra protease to get that virus primed to enter the cell. So, what they're saying is it's not a very effective virus on humans, at least. So subsequent work suggested that a glycan present near the S1 or S1, S2 boundary accounted for the lack of proteolytic priming of HKU spike and that its removal enhanced pseudovirus entry in human cells. Oh, yeah, this is important. So here they're saying that one of the reasons why that this HKU4 spike protein was unable to let the virus enter human cells was because there was a site very close to this to the cleavage site that it was difficult for proteases to work on it. So for example, here we can see our glycans on the spike protein. If this was a MERS protein, a MERS spike protein, and if specifically if it was one of the MERS spike proteins of the HKU4 line, then one of these sugars is going to be very close to the cleavage site. And if it's too close, or if it covers it up, it could make it very difficult for that cleavage to happen. And if it's difficult for that cleavage to happen, then the virus does not infect the cell. And perhaps one could assume that in bats, the bats have a very specific protease that is able to really dig itself in there, and that specific protease is completely lacking in humans. That is something that can be considered. Granted, if I remember correctly, the MERS and even the MERS-like spike proteins have a polybasic cleavage site, and so it probably also uses furin as well, but I would have to look back at that just to make sure. So, continuing on, the presence of a polybasic cleavage site that can be processed by furin-like proteases is a signature of several highly pathogenic avian influenza viruses and pathogenic Newcastle disease viruses. Strikingly, SARS-CoV-2 harbors a furin cleavage site at the S1, S2 boundary, which is processed during biosynthesis. So, strikingly, we have this very reactive cleavage site, and it's so active that it actually will break apart some of the proteins before the virus has even finished being produced in the original cell. Continuing, we speculate that the almost ubiquitous expression of furin-like proteases could participate in expanding SARS-CoV-2 cell and tissue tropism, meaning that they can infect more tissues than it would normally be able to infect relative to SARS-CoV, as well as increasing its transmissibility and or altering its pathogenicity. We previously suggested that coronaviruses use conformational masking and glycan shielding to limit recognition by the immune response of infected hosts. Similarly to SARS-CoV spike and MERS-CoV spike, we found that SARS-CoV-2 spike trimer exists in multiple distinct conformational states resulting from the S-binding opening and the trimer at the trimer apex. These structural changes are necessary for the receptor engagement of these three viruses and lead to initiation of fusogenic conformational changes. So after it binds, there's a conformational change of the trimer structure, and that is what helps allow the virus to enter the cell. In contrast, only S trimers have been detected for the 4 other human infecting coronaviruses, HCoV NL63, HCoV OC43, and HCoV HKU1, and HCoV 229E. Let me make sure I got this right. These structural changes are necessary for the and lead to initiation only. As HCoV NL63 and HCoV 229 are known to engage protein receptors through the S binding trimer, or S sub B, trimer opening is also expected to occur to expose their receptor binding motifs that are otherwise buried at the interface between promoters in the closed S trimers. Regardless of the nature of the receptor and the location of the receptor binding domains, removal of the trimeric S sub 1 crown is expected to be necessary for all coronaviruses to allow the large-scale S sub 2 conformational changes leading to fusion of the viral and host membranes. Collectively, these data underscore that S-glycoprotein trimers are found in highly pathogenic human coronaviruses appear to exist in partially opened states, while they remain largely closed in human coronaviruses associated with common colds. So what they're saying here is, and this was parroted, it's parroting what they said up here, is that for the less pathogenic coronaviruses, they are always found in closed trimeric states. It is very difficult to get them to open up. But for the other 3 coronaviruses, MERS, SARS, and SARS-2, they exist in partially open states. So they are pretty easy to get open, which could probably lend to it being more pathogenic. So based on the aforementioned data, correlating the binding affinity of SARS-CoV and HACE2 with the rate of transmissibility, viral replication in distinct species, and disease severity, we have hypothesized that the most pathogenic coronaviruses will exhibit S-glycoprotein trimers spontaneously, sampling closed and open conformations, as is the case for SARS-CoV-2, SARS-CoV, and MERS-CoV. The striking structural similarity and the sequence conservation among the SARS-CoV-2 spike and the SARS-CoV spike glycoproteins emphasize the close relationship between these two viruses that recognize human ACE2 to enter cells. This resemblance is further strengthened by our finding that the SARS-CoV spike elicited polyclonal antibody responses, potentially neutralizing SARS-CoV-2 spike-mediated entry into cells. We surmise most of these antibodies target the highly conserved S sub 2 subunit, including the fusion peptide region. Based on its structural similarity across SARS-CoV-2 and SARS-CoV, the lack of cross-reactivity of several SB-directed antibodies, and previous reports showing that the sera from SARS-CoV infected individuals target this region. So what they're saying is they have tested that if you make antibodies against the binding region, oftentimes it's not very effective at cross-reacting because these binding regions tend to mutate probably a little more quickly than the fusion peptide motif or the fusion peptide domain. So what they're saying is if you create a lot of different antibodies, a lot of those antibodies are probably going to target the fusion peptide, and by targeting the fusion peptide of one, that is most likely what's going to afford cross-reactability with the other coronavirus spikes. Continuing, we note that most SARS-CoV neutralizing antibodies isolated to date target the SV domain, and that several of them recognize the receptor binding domain and previous receptor engagement. As the SARS-CoV-2 and SARS-CoV SV domains share 75% amino acid sequence identity, future work will be necessary to evaluate whether any of these antibodies neutralize the newly emerged coronavirus. These findings also indicate that it might be difficult to distinguish exposure to SARS-CoV-2 from other SARS-related coronaviruses in serological studies using the S ectodomain trimers, and that specific assays will need to be designed. So here is something that I have been worried about, that with our antibody studies, is that if we are doing antibody studies to see how many people got infected with the coronavirus, then we need to be very careful, because if there are different coronaviruses that are also creating antibodies within the people that they're infecting, we got to be careful to make sure to account for the cross-reactivity of these antibodies. So we don't want someone to test positive for antibodies for the SARS-2 coronaviruses, or the SARS-CoV-2 coronavirus, when in fact, maybe they just got infected by a completely different coronavirus that is a lot less pathogenic. So they might go out thinking that they're completely immune. They might go out thinking, okay, everything's great. But especially if those antibodies are only targeting some of the, if the cross-reactivity is not perfect, then they might get infected later thinking that they were okay, and they end up getting hurt in the process. So continuing, our results provide a structural framework to identify conserved and accessible epitopes across S-glycoproteins that will that will support ongoing vaccine design efforts. Finally, elicitation of diverse polyclonal antibody responses might prove key in light of the diversity of viruses circulating in animal reservoirs and in preventing the possible emergence of viral neutralization escape mutants. So here they're saying they're worried. We got to make sure, if we want to be a little bit better at protecting ourselves from a reemergence of a different coronavirus, or at least a different mutant of the current coronavirus that we have going around, polyclonal antibodies might be a better solution. So especially if we can get them from mice and purify them from mice and then inject them into ourselves, that would be an example of using polyclonal antibodies that could be cross-reactive, could protect us from many different types of coronavirus spike proteins. So we're done, finally. And yeah, here they're just talking about the fusion peptide and how the fusion peptide area is a little bit more, or quite a bit more conserved than the S-sup A domain and the S-sup B domain. So as you can see, this is the top view, or this area. So this area is a lot more, has a lot more variety, probably because it's more exposed, and probably changes more to avoid antibodies of the different creatures that it runs into. But this area that's a little bit more tucked away, so here's the fusion peptide, here's the fusion machinery, this is a lot more conserved, and so we presume that if you do have antibodies that work on multiple coronaviruses, they are going to be working in this area. So thanks. I think that is everything, so please subscribe. The next video that I'm going to be making, I'm going to change gears for a little bit. So I want to talk about a different strategy that some coronaviruses can use. So I want to talk about how coronaviruses might be implicated in causing things such as depression in people and hormone dysregulation. And one of the ways that viruses are able to do this. So we were talking about a lot of proteolytic cleavage that is going on within these viruses to help process them. But a lot of the proteins that are cleaved can have different effects on the human body, especially, and this is one reason why we want to work for a vaccine rather than getting infected with the virus themselves. These pieces of proteins, if you make antibodies against them, some of these protein pieces will function like hormones in the body. And what you do not want is to create an antibody against one of these pieces of protein that look like one of your hormones. Because what will end up happening is the hormones that you produce yourself, some of them are important to survive or to be happy and healthy. Um, if you make antibodies against your own hormones, that is going to dysregulate your hormones. So I should back up a little bit. If you make antibodies that can attack the proteins that are mimicking your hormones, those same antibodies are going to attack your own hormones as well. They might not work as well on your own hormones, but they might work, and that can cause dysregulation. And so what I'm going to do is I'm going to make another video talking about how this is a strategy that some viruses employ to help do immunomodulation so that the viruses can live a little bit longer within the body. So with that, thanks for watching. Thanks for listening through all of this. I will hopefully get another video up within the next few days. So cheers.